Let me invite Jaime Prohans, who's going to co-chair the session with me, the second session, and it's about EU projects. Again, the first speaker is Tony Granell from SIC in Valencia, and he's going to talk to us about two projects, one Horizon 2020 and one Horizon, both of them Horizon 2020 actually, on tomato genetic resources.
Thank you, Giovanni. I will be rather fast because I have a flight to catch and also because of the tight schedule we are already in. So these are two projects. One is still running and the other was finished, both dealing with genetic resources and in tomato. So the large title of each of them, the oldest one, TRADITOM, is about traditional tomato varieties and is to introduce science into defining what traditional varieties are. And the other is more like this is a RIA, a research and innovation action, while HARNESSTOM, that's still running in the third year now, is about how to use the knowledge we have about tomato diversity to engage with companies to produce new pre-breeding materials. Okay, so they are very good funded. I mean, one was more than 4 million, the other is 8 million euros, and the call was under the traditional resources in the case of TRADITOM, and the innovation action under which HARNESSTOM is included has to do with genetic resources and pre-breeding communities. The objective in relation to genetic resources of these two projects are different. In TRADITOM was about information and conservation, cataloging and breeding and valorization. And we did lots of activities in collaboration with farmers and breeding companies in the first case. In the case of HARNESSTOM, is an innovation action. So one of the main pillars was to build a database and visualization and analysis tool. It was a requirement call. And we tried to organize this for tomato genetic resources. Was also another important activity in HARNESSTOM is to breeding tools to speed up breeding, it's one important approach. And then in the case of breeding, we target different aspects like disease resistance, or climate change, or fruit quality, and with collaboration with companies, but also as participative breeding. And both of them have large consortium of close to 20 research institutions. And in the case of TRADITOM was what they would call as a multi-actor approach. So we got involved many other companies, association of producers, consumers, and so on. So is the case of HARNESSTOM. Okay, so the idea in TRADITOM, I will be talking about TRADITOM in the next few slides, is there is a large phenotypic variability. We wanted to understand the genetic basis for that, and then we produced a detailed catalog of each of the more than 1,700 varieties, traditional varieties, collected all over Europe and stored in gene banks in Europe, APK, Bari, Comab, and some universities. And eventually it was published in a couple of papers. We genotype and phenotype extensively this collection. This is a map where all this germplasm derives from, mostly from the southern countries in Europe. And we phenotype 71 traits, and we ran more than 10 field trials of these 1,700 accessions of tomato. It was a collaborative effort over these 4 years. And then scientists were interested in finding the genetic basis of some of the traits. So we applied a lot of analytical methods to try to get the information out of these huge data sets. Eventually, we end up with a lot of markers associated with traits, which is very good. We also produce a core collection out of the 1,700 varieties that included 227 accessions, traditional varieties plus 50 varieties from companies. And we ran parallel experiment with all of them in 4 different places, in Perello, in Viterbo, in Avignon, and in Israel. And we addressed the question of stability on different agronomical traits of importance for tomato. And we end up also with identification of genomic region that had to do with stability of all those traits, agronomic traits that are of high interest. In the case of, but again, we did a lot of activity with farmers. We integrated the information we got with them. All the cultivations we did was not in the university facilities, was in close interacting with them. In the case of HARNESSTOM, the approach, you know, in the case of the call where HARNESSTOM fits, the idea was to enhance management and use of genetic resources and implement global commitments in this area. So the approach we took, it was first of all to produce a one-stop shop for tomato genetic information, and also to develop tools to speed up breeding in some of the activities that we have, but also to have targeted alliances between the academia and seed companies and farmers. Okay, and these were different work packages, and they were like ring-fenced type of pre-breeding sub-projects. But we also have participative breeding, in which we involve academia and farmers. Okay, and we were very active. Just we'll go a little bit through the different activities that are quite well advanced. The first was the compilation of literature, the information databases, our information about climate traits and genotypes associated with climate change, with emerging diseases, and with fruit quality. Then there was, at the beginning of the project, it was clear to us that there were some emerging diseases in the case of tomato, like is the brown fruit rugose virus, and we needed to screen population to find new sources of resistance, but also for the new daily virus and Cadosporium fluvum, that there were strains that overcome the current resistances, and there was a lot of activity in this. And so what we did in this ring-fenced type of approach with each individual company is to select a number of traits with specific materials from elite lines from the company, and to carry market system plant breeding activities with them. Then we are now at the phase in which we are also parameting some of these resistances, and then in the last year we will perform to evaluate these materials in different environmental conditions in different places throughout Europe. In the case of the participatory breeding with farmers, the first activity was the definition of ideotypes by farmer consumers and influential chefs. We have also a lot of involvement in influential chefs in the project. And another activity was produce another repository of another collection of varieties that were assessed last year in Bulgaria, deriving from information we have from previous youth-funded projects. And also the more specific part of this participatory breeding was to do a lot of citizen science, and also to do a number of breeding projects with the farmers. In one case was to study the evolution of land races in order to diversify for color and quality. And this is quite well advanced. In the part of tools to increase the speed, efficiency, and precision breeding, we have been able to reduce generation time using physical and chemical methods and genetic methods. We also have identified regions of some wide relative that have the ability to increase recombination rates. We are very active to add additional genotyping and phenotyping, genotyping information on collections of material that were not so fully characterized. And then we are developing genome-oriented selections to increase breeding gain. And this is activity some of the partners in the group. Okay. And then we are compiling let's say, among the different partners, materials and populations that can be used to increase breeding efficiency. The idea is that with this project, first of all, we have a strong alliance with the different stakeholders from using genetic resources from breeders, of scientists, also farmers and public at large. And then the idea to build a public repository of all the information that is linked to the germplasm banks, then tools and materials for precision breeding, and then to demonstrate that we can do this in a number of pre-breeding targeted projects with the companies. I will just go through a little bit of the database of where we are setting up for all this information, and that the idea is that information that was produced in a number of EU projects that is not yet centralized in a single repository for information. For instance, TomGem, Bresov, TomBres, or UTP-SOL, TRADITOM, HARNESSTOM, we will have it in one single place. And so far we are on the right way to do this, and we have a first version of the HARNESSTOM databases. They have different models. One model has to do with experimental details about the crop, the program, the trial, the study, the experimental design, you name it, a number of them. Then we have the germplasm model, where you have the accession, the taxonomy, the synonyms, the accession attributes, etc. We have a phenotyping model where we introduce all the data that has to do with the traits, with the observation variables, with so. And then we still have another module for the genotyping. We have what genotyping platform was used, what software, with the markers, the bioproject ID, the variant, and so on. And still we have another module to having all the variant catalogs, whether it was single or multi-environment QTLs, the clone gene in case it was cloned, etc. What we have so far until now, we have been able to upload results from 3 programs in TRADITOM, TomGem, and all mapping that correspond to 66 trials and to 24 studies. These represent more than 2,000 different germplasm, correspond to over 1,900 accessions with all the information about passport, attributes, and so on. We have more than 10,000 images. We have crosses, mapping population, and so the pedigree of a number of them. The number of variables in the phenotyping module spans over 170 observation variables, 872 metabolites in the fruit that correspond to health-related or sensorial perception of the food quality. We have more than 4,000 observational units, in total more than 90,000 phenotypic variables on the database, in the current version of the database. In the genotyping module, we have 4 genotyping platforms, more than 100,000 markers, and so on. And the case of variant catalog for QTL studies, for GWAS studies in a large number of cases. And the idea is to start to, this final year, to introduce all the data related with other EU projects like UTP-SOL and so on. Okay. The data will be public. We are still building the database and populating the database with information. And as soon as the paper come out, everything will be public. All the data related to TRADITOM that they mentioned, the numbers they mentioned, the data will be released soon. It's already open. And the way that we understand this database is that it's very easy to exchange information and to obtain information either through a web application or from API access. And this is the way it looks so far. It has a number of features that you can go through, the location of the accession, a number of pictures, and we are very happy so far. The database is until now is stored in Toulouse, under this, you can get there with using this URL. And in the next few months, we will be more than happy to include studies and new studies or results from that we already have available, but have the time to introduce the data, and then to habilitate a download tool, QTL by QTL epistasis, a little germplasm view, and so on. That also to transfer and connect HARNESSTOM with phenome networks. This is a platform more defined for interaction with breeders in companies. And then also with Sol Genomic, that so far has been the standard Sol genomic platform for Solanaceae. And also interconnect with Tom Express, where you have a lot of gene expression data related to basically for fruit and plant expression data, and also to expand the visualization and analysis tools. And the idea is that this is to connect this with the seed bank, so there will be a specific tool that once you get select an accession based on the phenotype on your analysis, you can request the seed going through the specific seed bank where the accession is stored. Okay, and we want to, of course, to include additional data, populate the database with more data. So this is the acknowledgement of for the EU and the people who contribute with data, a lot of work during all these years. And sorry I went through so fast, but we are late, and my flight is probably late. If I arrive on time, I will be late if I arrive on time. So, thank you.
The EU G2P Sol project. It was a 6-year project, started in 2016. It went on until end of 2021 with 19 partners from among which 13 gene banks from all over the world. As you can see, we have big gene bank in Taiwan, the World Vegetable Center, and big gene bank in South America, SIP, and then several European and from associated countries. And the project was focused on the 4 main Solanaceae: tomato, potato, pepper, and eggplant. And these were the accessions in the different gene banks. You can see we have about 64,000 accessions of the 4 major Solanaceous crops, of which about 14,000 wild relatives. So this is about, it's a sizable amount, about 40 to 50% of the active worldwide accessions of these 4 crops. So it's very representative of the worldwide biodiversity. And when we started, I like to compare it to what you find in the Biblioteca Angelica in Rome. It's a very nice collection of ancient books with a sketchy catalog. So only experts can find the book chapter they're looking for. So first of all, we collected the passport and phenotypic data and images that were scattered around different gene bank information systems. There was a lot of manual curation, and to make them homogeneous, especially the quantitative phenotypic data that were scored with different scales. And we have now in a centralized database here, the G2P Sol Gateway, about 56,000 passport data for 56,000 of the 64,000 accessions, phenotypic data observations for 28,000, and images for 17,000. So you can all access them freely at this address. And this gateway, I have no time to go through it. It can, besides phenotypes and metadata for field experiments, it can host, it can allow several functionalities, design field experiments, randomized block field experiments, establish ancestries by pedigrees, allow basic genome association wide studies, and it can host also genotypic data associated with the accessions up to 200,000 markers for each accession. So it's a very flexible database. It's privately hosted. It's mirrored on the public partners of the project, and it will be maintained until the end of 2026. So the second step in the project was to barcode with high-density methods about 40,000 of these 64,000 accessions. And the methods we used were GBS for pepper and eggplant, and SPET for pepper and potato, and SPET for pepper and egg, and for eggplant and tomato. I don't have time to go through the technicalities. Suffice it to say that this is a non-targeted method. This is a targeted one. And you can see this in the efficiency of the methods. You get about, in pepper, you get about 2,000, 3,000 high quality SNPs for each 2 million mean reads per individual. To get much higher numbers of high quality SNPs from eggplant, you need much lower number of reads per individual with SPET, and this is because it is focused on highly variable regions. This was used to correct the taxonomic assignments in gene banks, correct mistakes, and to spot duplicates. And how do you spot duplicates? Basically, you genotype the same genotype over and over again. You establish a threshold diversity of the same genotype, and then you score the whole gene bank collection, and you say, okay, below this threshold, this is considered a duplicate. Above this threshold, this is considered a non-duplicate accession. And this is, of course, since it's done with a method that does not address all the sites on a genome, it's a tentative classification. Then you have to go back to passport data to confirm that indeed the candidate duplicates that you find in different gene banks are similar to each other. And what does this allow you to do besides reclassification and spotting duplicates? Since many of these accessions are georeferenced, it allows you to reconstruct the domestication and expansion history of a crop. So for pepper, for instance, this is a paper that was published in PNAS in 21. The domestication center was well known, was mid-America, and then we found several expansion routes going to South America, then through the around Africa, probably by the Portuguese from Brazil. It went to the Middle East, to the Far East. We found a second trans-Pacific route from Western Mexico to the Far East, and that was probably mediated by Spanish galleons that went all the way from Western Mexico to the Philippines, etc., etc., Silk Road. And you can make a map of the expansion of the different groups and of the characteristics that were selected in each of these geographical areas. And you can make the same for eggplant. And for eggplant, there was a surprise. There was a lot of debate whether it was domesticated in just one place or two places. We find very strong evidence for two-side domestication. In Southeast Asia, that probably gave rise to the Chinese, Japanese, and Far East accessions. And in India, that probably gave rise to the Middle Eastern and Southern European accessions, probably brought to Europe by Arab traders in the late part of the last millennium of the 900 AD. And then you can use gene bank data, phenotypic gene bank data, also to make GWAS, genome-wide association, over phenotypic data collected by gene banks. And this is like a very large experiment with thousands and thousands of accessions. The different gene banks have scored. As I said, it requires a lot of manual curation, but you get signals. So this is a well-known gene for pungency in pepper, is capsaicin synthase. You find it again from genotyping and gene bank pungency data of the 10,000 accessions that were scored in this experiment. And you find new regions, like this one is a very interesting region that controls the number of pedicels per axis, so the number of fruits per inflorescence. And it's a completely novel region. You find it from historical gene bank data, and there is a possible candidate gene, is a homolog of a tomato gene that controls compound inflorescence. And then the step 5 that took an enormous effort from the consortium was to select, based on this very large scale genotyping, accessions that were representative of this worldwide genetic diversity, issue phytosanitary certificates for all of them, and exchange them between gene banks to establish these core collections. This was like an enterprise that took about 1 whole year for the 3 seed propagated crops. And it never ended for the tuber propagated one, for potato, for which this was just not possible. There was no enough resources in the project to establish a unique collection between South American and European accessions. So we ended up making 2 different collections. And so I'm referring here to the 3 seed propagated ones. This is the number of initially genotyped accessions. Some of them were preselected by the partners as reference accessions. The target total number was this one, and this was the rest of the rest of the accessions besides the preselected were based on genetic distances to just make sure that the whole diversity was represented in this core collection. And these are very important resources that are producing an incredible amount of new data because they are representative of the worldwide diversity. And they have been extensively phenotyped in different fields across the globe. You can see all the experimental fields, and these are all the data that we have collected in these different experiments. The resequencing was completed for eggplant and tomato, is underway for pepper, and the GWAS analysis is underway, and it's producing hundreds and hundreds of novel quantitative trait loci. So how the main question, and that's how I'm going to leave it, is how are these resources going to be immortalized? So the core collections, individual accessions, are available from participating gene banks under SMTA. But gene banks depend on funding by national governments for their future operation. That's a problem. The data are in the G2P Sol Gateway. It's a very nice one. It has several functionalities, but we have resources to maintain it only until 31st of December 26. So after that, these data are on their own. And this refers to what about long-term conservation?
Right, like all the other speakers, I'm going to start off by saying I'm going to be very brief and I won't keep you long, but I want to really do justice to the partnership we had in Farmers Pride. So Farmers Pride was an EU-funded Horizon Europe project that finished a couple of years ago now. And essentially, it was about developing methodologies for in situ conservation, both for land races and for crop wild relatives. But I'm going to focus, I can't cover all of it in a short presentation, so I'm going to focus on the crop wild relatives side. But before I start going into the actual project itself, I wanted to give a little background. So this is my analysis of where I think we are with the role of gene banks. So gene banks, I looked quite a long time actually to find a definition of a gene bank. And this is a definition I found back in 1991 for a gene bank. So gene banks, their role is to maximize conservation, characterization, documentation, and use. Use is the important link here, for the benefit of humankind. So in other words, it's not just devoted to putting material in a gene bank. It's the broader focus on conservation, characterization, and documentation of all genetic resources. And you can see basically the process here, the materials in nature or on farm if it's land races. It's located, sampled, transferred if it's going into a gene bank. You do the standard processing. It's regenerated periodically. It's then characterized and goes out to the user community, breeders. So there's this very important link. And I emphasize that link works. It works perfectly. But the slight limitation is there's not enough material going to the breeding community. So increasingly, you see publications talking about the lack of sufficient access to diversity, restricting plant breeding outcomes themselves. In other words, the breeders want more diversity, they can't get hold of it. So what we should be focusing on, the hold-up, the bottleneck, is actually getting more material into the gene bank and available to breeders, or available to breeders in some other form if possible. Now, at the same time as that requirement for more genetic diversity, you've also got erosion and extinction of genetic diversity that exists on farm and in nature. And if we look at the IUCN Red List assessment for crop wild relatives, there are 2 or 3 papers that have done large-scale red listing, and the percentage of threatened crop wild relative species is between 16 to 35%, depending on which one of these publications. Then if we do an analysis of the material held in gene banks of crop wild relatives, about 1/3 of the species in gene banks of crop wild relatives are not there at all. There's no material for 1/3 of crop wild relative in gene banks. About 1/3 are poorly conserved. In other words, they don't maintain the genetic diversity, so it's less than 10%. And 95% of total of all crop wild relative material is not adequately represented in gene bank sessions. Then we look at the money that's recently been going into ex situ collecting. So this is the Trust project in their summary paper at the end of the Trust project. And I put here a budget of about 8 million. The paper actually doesn't give the amount of money that was spent on this project, but I think vaguely it's around 8 million. So forgive me if I've got that slightly wrong. But as part of that project, over 5 years, they collected 4,500 accessions of 28 crop wild relative taxa from 25 countries. 8 million, and they collected 4,500 accessions. So if we really want to fill these gaps, my point is we can't rely just on ex situ alone. We should try and be promoting in situ. And if we have a truly complementary approach, both ex situ and in situ, then we can significantly improve or the amount of diversity that then is available and meet this bottleneck for breeders. So we then produce this. So complementary conservation is what we should be aiming at. And the point I'm making is if we do that, we can almost double, I think, the amount of genetic diversity that's available to breeders. Okay, so this is then the revised model for what we're proposing would happen. So instead of, again, making the point that they are no longer gene banks, because essentially what they're doing is they're leading the activities for in situ and ex situ. So I think there's an important distinction. So that's why I prefer to call them genetic resource centers. And we have material going from the wild, from on-farm, via ex situ, via in situ, into the gene bank. So historically, when we first started talking about in situ conservation, we all assumed that the in situ resource maintainers would be supplying the material themselves. But after working on this for 30 years, I still find it impossible to find an in situ maintainer that knows enough about the CBD, knows enough about the appropriate legislation to provide adequate access to the material they're actually conserving or holding on farm or in situ. So I don't think it's possible to do it that way. So the way to do it, as I see it, is we get each in situ population, whether it's maintained in a protected area, whether it's maintained on farm, to send a sample, a backup sample, to the gene bank or genetic resource center, as we now call it. And then that material, they are then in charge of making the link with the users, because we know that that element of the gene bank works extremely well. They already have that process. So that way we can then open up all that in situ conserved material. So this is the background to Farmer's Pride, what we were actually working on, trying to promote that link between conserved material in situ, on farm, crop wild relatives in genetic reserves, and then link that through to the gene bank, and then link that through to the breeder. So that's what we were working on essentially. So if we look here at the steps, genetic resource centers, as I said, engaged in situ planning. So they're planning, they're helping plan where you're going to do the actual conservation. They're actually engaging in situ conservation. This will incur a cost. But if you look at the benefit of doubling the amount of material available to breeders, then the relatively small cost is significantly outweighed by the access to the additional genetic resources. So if we want to do something about feeding adequately the 850 million people worldwide who are currently suffering malnourishment, we want to do something about them, then this is the appropriate option, I think, to take in terms of genetic resources. Right, let's focus more then on Farmer's Pride for the last few slides. Okay, so Farmer's Pride was, as I said, a Horizon 2020 project, networking, partnerships, tools for enhancement of in situ conservation of plant genetic resources in Europe. 3-year project, budget of only 2 million euros. The aim was to improve best practice and tools for in situ conservation, to look at this link between in situ and ex situ, to help influence policy, and our goal, and this is perhaps me being slightly ambitious in setting the original goal for the project, was to actually set up a European network for in situ conservation and sustainable use of genetic resources. So for crop wild relatives and for land races, we have a network across Europe of where that material will actually be conserved. We had 19 partners and 19 what we call Farmers' Pride ambassadors. So the ambassadors were national representatives, which would help us in terms of developing national in situ policies and national reserves and on-farm locations. So, as I said, the primary goal was setting up this network of sites, permanent system of in situ conservation for plant genetic resources across the whole of Europe. And this involved localities where you had populations of crop wild relatives or land races maintained on farm to an agreed standard. And I'll be talking about standards this afternoon, so I'll come back to talking about those again. And the people. So not only do you have the resource you're conserving, you have the people that are maintaining them. You have the genetic resources centers because of making access to the user. You have to have this link between each in situ site and the people who are going to, where the material is going to be deposited and then passed on to the end users. So you have to have that link. And then other stakeholders like breeders in terms of defining what is the material that breeders are most interested in, and then making sure we can get that material from in situ. So what we could stop, we could say, okay, we just conserve the material in situ, and then that's it. We don't have this link to utilization. But remember back to that definition of gene banks, there has to be an explicit link between conservation and use. And unless in situ has the link through to utilization, then it won't be equal to ex situ, and it won't get funding in the long term. So the only way we can get long-term funding for in situ is to make sure that in situ to utilization to breeders and other farmers works. Okay, so some of the products we developed are, we helped develop further cat phytogen, so we could do GIS analysis of various kinds. We produced a population lookup tool where you could look up any protected area in Europe and find out which crop wild relatives were found in that particular location. We developed in situ management plans, and because Bella's sitting at the back here and is going to be grumbling at me if I don't mention it, we also definitely produce community-based guidelines for maintaining material either on farm or in garden. So we produced a lot of useful tools in that sense. But essentially, and again, I'm focusing here on crop wild relatives. Essentially, if we're working on crop wild relatives, we have two locations they can be conserved, either in existing protected areas. And why are we doing it there? Because they already have a long-term ethos for conservation. So we're utilizing those management regimes to actually more actively conserve wild relatives. Or I think increasingly, we will use sites away from traditional protected areas where we have, say, round the edges of farmland. And this is the sort of support that will come from government stewardship schemes, where you're being paid to maintain crop wild relative populations, or in the landrace case, you're being paid to maintain traditional varieties in this sense. So those would be other effective conservation measures. And then I wanted to make the point here about the link of the different levels. So if you're doing conservation of this kind, it can be done at a local level, it can be done at national level, and it can be done at regional and global. So if you look at this simple pyramid diagram here at the base, we have all crop wild relative and landrace populations throughout the whole of Europe. Some of them will be included as national actively managed populations. Some of those will be included at a European level, being given European priority, and then some will become part of a global system. And then this model comes from something that predates Farmers' Pride, which was the ECPGR concept for crop wild relative conservation. So here, essentially, what you have are the basic methodologies. So the green circle with the point in the middle is actually, what's that? The national. So that's the national process for identifying crop wild relative sites. And the blue circle, which has similar information and uses similar processes, is the European level. So what we're saying is when you're actually planning in situ conservation of crop wild relatives, you're actually combining information from different countries, a bottom-up approach, but also a top-down, because you're also planning and identifying sites of European importance. So to actually set up a European network, you need to combine those two and have this integrated European conservation structure. And the same process we thought roughly would work for land races as well, maintained on farm. Now, I'm not going to explain this diagram here because I'm going to go through it in detail this afternoon. But just to say, this is the means of the gene bank or the genetic resource center actually taking the backup samples and how they're actually stored in the gene bank. And then we produce minimum standards for individual reserves, how individual reserves might form within a network, and then the actual process of adding individual sites. So first of all, you would have an area in your country which has a high population or a target population, a lot of diversity of crop wild relatives or land races. That's then discussed at a national level and nominated by the national coordinator to the European network that would be established. If it passes certain criteria that I've just mentioned, that we've also outlined, then it becomes a member of the European network. And then periodically those membership criteria are tested, and you can be, if you don't meet the requirements, you can fail and have to go back and go through the process again. So it's a simple methodology for how making sure the material within the European network maintain currency. Okay, so as I said, the main point, this is the last slide, the main point of Farmers' Pride was actually setting up this network. And to be frank with you, we didn't set it up, but we got bloody close. And we developed a lot of the tools and protocols and procedures that would enable us to set up the network in what we hoped would be a future project that would, and I'm sure this will happen in time. But what we did end up was very encouraging. So this slide here, after we've been working on this, as I said, for 3 years, we put out a call across Europe for sites or managers of sites where they had either crop wild relative or land-raised populations. And we asked them to say whether or not, if we set up a network, they wished to join that network. And we got all of these sites showing a complete coverage of Europe and a wide range of different stakeholders who would be involved. So this to me is encouraging.
Thank you, Nigel. And now it's the turn of Bruno Mezzetti online.
Thank you very much. Well, again, first of all, thank you very much for inviting me. Sorry not for being here, but it was difficult for me to join this very interesting workshop. I was following some of the presentations this morning. Well, I have a short time for presenting what is the breeding value. Breeding value is an Horizon 2020 project that is linked to the other projects that were approved inside the genetic resource program included in the Horizon 2020 project. So we focus, our team is on trade breeding strategy for obtaining new resilient added value berries. So we focus on berries. We focus on berries because we propose 3 important berries, strawberry, raspberry, and blueberries, that are having a quite high impact at international but also European level in terms of cultivation, but economic value, and also in terms of rural development, because these small fruits are giving to the small company a good possibility of income. So that is considered an interesting opportunity for the rural development. Strawberry is the most important, followed by raspberry and blueberries. The interesting aspect of berries that is they are keep showing an increase in terms of berry consumption, still estimated at plus 3–4% as annual rates, particularly for strawberry, but for the other crops, for raspberry and particularly blueberry, this percentage is increasing even much more. The blueberry is based on the fact that we want to create a long-term European networking with a specific activity on research applied to small fruit. I'll say it's taking the 3 crops as a case study. We have 20 partners, including from 8 different countries. In reality, then we also had 4 open calls that allowed to include additional partners, in particular private company. The project started in January 21 and is going to end at the end of 24. We are now, however, planning to ask for an additional at least a semester, so an extension. I don't know if it will happen, but there is a aim. So just to have you a distribution of the partners at the European level, from the west to the east, from more or less central, south of Europe, north of Europe, going up to the Scandinavian countries. These are the partners, and as said, these are the different companies, mostly small companies, breeding companies and nursery that have an interest on berries, propagation and cultivation. So that was the approach to enlarge the partnership or the access to the data that we are going to generate in the breeding value. The idea of the breeding value starts from the concept that breeding in some way have, at least for berries, a strong impact in reducing the allelic diversity. And actually, the idea started from a paper published by a Spanish group in 2009, demonstrating that particularly for strawberry, in 60 years, about 60 years, there was a decrease in terms of allelic diversity of about the 35%. Of this 35% occurred practically in 10 years. This were practically the breeding for strawberry became more intense, and so there was a very strong reduction of number of cultivated variety that replaced the more traditional cultivated variety. So, starting from this concept, we try to identify what is the difference between the genetic resources that are available. So, we prepare practically 5 categories of different varieties, plus a category that is a breeding population. But about varieties, we have the first category that are the old varieties, germplasm, that is being released, identified before 1960. So let's say all germplasm, some varieties that were produced by breeding, but in reality, really, this represents a very large diversity that we have in the germplasm collection. Then varieties that were released from the 60s to 2005. This again, so breeding, so when breeding became more intense, and but still based on small breeding program, based on the use of genetic resource and so on. And then more advanced variety that are practically in the last 15, 18 years. So the very, let's say, top now commercial varieties are recognized as important global variety, important at global level in the strawberry market. Then we have the category of the pre-breeding material. So we have what is available in the different breeding programs that are included in this by partners that are having partners included in this project, and also remembering that we have the wide species. In this case, regarding strawberry, we are speaking about Fragaria chiloensis and Fragaria virginiana, that are the two representative cultivars that generate the Fragaria per ananassa, so the cultivated strawberry. And I say the 6 categories that are the breeding population. Well, I'm presenting this for strawberry. That is more, it was for us more easy because previous project allowed us to identify already a wide range of germplasm and already characterize a lot of this material. So in the breeding value, we take data that were available thanks to already previous project already handled. But the same classification was done also for raspberry and blueberry. What to do on this genetic material? So the project came to first to identify and characterize the genetic resource. So that was for package 1. Apply on this new genotyping technology based on, so to validate new markers to apply on berries, because still it's not so well common to apply on berries genomic selection, and marker trait association, again related to the marker. Phenotyping, looking at the different new techniques that can be used for phenotyping the plant. So not only field phenotypes on quality, resilience, but also new tools for based on image analysis, for example, for defining plant architecture, for identifying plant diseases or specific traits related to the fruit quality. Metabolomic analysis, again, for more deep analysis of the fruit in terms of fruit metabolic contents, and the strict link to the consumer was thought by involving a group specialized on panel sensory tests. So we include in the project this, we will see on our package 4, the sensory analysis that is related to the consumer study. So based on this, on the survey, on focus group, definition of sensorial profile, consumer test, and so on. Then data management, EP management, that is also an important issue. Last March in Dresden, we had a workshop related to intellectual property management. We found that it's really important to keep the discussion between, for example, CP4, EPVGR, and also FAO that have 3 important frameworks related to the property of the management of the genetic resources. Well, the scheme is based on these different trials. So genetic resource.
Bruno, I'm sorry, could you?
I speed up. Our large variability is for strawberry, less for the other. Genomic studies are more advanced, and preliminary data we have classified already the difference between the old variety, the different type of modern and new variety, and new cultivars are differentiated between US and European product. Specific data are already on genomic selection for disease resistance. This is a platform for the phenotyping, so different technologies that we use, and this is the platform for the sensorial analysis. I go fast. And this is a platform for the data analysis. Then we try to improve information on berry research and to align information on berry research to the information then are available at the EU level, international level on plant genetic resources. More detail on the work that we are doing are already available on paper published last year on the project. Sorry if I was taking some more time. Thank you for your attention.
Thank you very much, Bruno, for this very interesting presentation. And we have a small change in the schedule. Okay, so Ivan, the floor is yours. You can share your screen.
Yes, great. Thank you very much. I guess if I do this, you should be able to see it full screen. Right. Okay. So let me go back to the first slide. Thank you very much for inviting me to present our FORGENIUS project. So FORGENIUS is an H2020 Horizon Europe project started in 2021 and lasted 5 years. So the project is dedicated to the conservation and the description of the forest genetic resources in Europe. The goals of the project are to increase the quantity and quality of information on the diversity, including phenotypic and ecological diversity and resilience of European forests. The other goal is to homogenize the structure of information that is collected in the different countries in Europe, across species and across collections. I'll be back to this in the next slides. And then converting the information that is collected into prediction and management tools, and of course making all these available to the end users and the public. So this is the measles map of Europe. In that represents the small dots, small red dots represent the places where conservation is actually taking place. And actually in forests, the situation is a bit different than in crops, because we don't actually have, with few exceptions, ex situ conservation. So everything you see here is in situ conservation, meaning pieces of land, forested land, where the genetic resources are conserved. We call them genetic conservation units, like in the general context of conservation of natural resources. And currently actually more than 4,000 of them that cover more than 100 forestry species. So the forest genetic resources are conserved dynamically, meaning that you are not conserving individual genotypes or clones, but you are actually conserving the dynamics of natural populations that allow you to maintain the diversity within the forests. So how do we go about this? I'm going to present the project, but not the results, because it would take too long. But we are now halfway into the project, and results are actually coming from each of the areas I'm going to cover in my talk. So the fact that we organized conceptually the project into what we call the pyramid of research fields and data sets. So the larger blue block that you see is based basically on remote sensing. Remote sensing allows us to collect pieces of information from the ecology, physiology, and so on, biomass, etc., from all the GCUs, so in a single shot. The smaller red block is a subset of the GCUs that we are going to characterize genetically by genotyping with the most advanced method that we have in each of the species. And we will focus here on 20 species and covering 10% of the conservation units. And then the smallest green block is the harder to come by type of data, that is the physiological, ecophysiological characterization of the stands. But then again, all these blocks or layers are connected together through modeling. So that means that based on the data we get from the small green block, we can extrapolate to a larger amount of a larger coverage of the collection through modeling based on the observations. This is just an overview of the partnership. We have 19 partners in 10 countries, and with a mixture of research, academic organisms, international policy support, and an industrial partner in the consortium. I'm just going to very quickly through the structure of the project by the work packages. But basically, you have this green left side that is producing the data and the results, and the purple block, which is actually organizing the new data sets and turning them into a new information system. We already have an information system about the network of GCUs, but it is quite rudimentary, and it requires to be pumped up and made way more informative for the users. And then around you have a sort of circle or square, actually, with the interaction with the end users and engagement with the end users that actually occurs from the beginning of the project, because we need to make the output of research match with what the forest genetic resource managers are used to use as tools to survey and monitor genetic diversity. So I'm just going to give you a brief overview of the methods we use, the approaches. The first kind of things we do, as I said, is to use remote sensing and modeling, and that allows us to have a full coverage of Europe, and to characterize our forest plots, which are our genetic conservation units, using spectral data to feed ecological models, and then use ground data to validate the models. And this way, for a given species, we go and actually characterize only a subset of the GCUs, but then we extrapolate to the whole set of the GCUs for that species, which is very handy. Plus, this allows us to update the models every time that there is a new production of satellite data that we can use. Another aspect is to look at the short-term resilience of the genetic conservation units to extreme events like drought events, heat waves, wind throws, and whatever you name. So in this case, we are looking in particular to the water, to the hydraulic traits and the water relationships, which seems to be a very major thing in the next few years. And so we are focusing on looking at how individual trees can react and what their resilience is to these events, and then scaling up to the whole forest based on the single observations, and then modeling the long-term, short-term response of individual trees based on their traits. And so that will allow us to have an overview at the European scale of the resilience of these objects. Another way of looking at it is what you could say can call adaptability, so the evolutionary potential, which is a long-term resilience aspect. And in this case, we will apply the classical quantitative genetics, quantitative evolution.
I'm sorry, 2 minutes.
Yes, sure. It's almost done. And so we will take this approach that will allow us to extrapolate long-term adaptability of the forests. And this will globally will produce multidimensional indices of adaptability and resilience of the forest, and this will be made available to the end users, which is basically the network of forest management systems throughout Europe, by restructuring the current database and providing the new data in a flexible way. And this will be provided to the users, basically mostly in the same way you could think about GenBank, something that can be queried and you can take information out. And so just to go to the end, the take-home messages are that what we want to do is to homogenize the structural information across countries, species, and conservation units, converting information into predictions through modeling and management tools, and making these tools available for managers, but also the general research community, including you, of course. And the important thing is that this is a generic approach to the characterization of the genetic resources. In this case, it is applied to Euphorgene network, the network of conservation of forest genetic resources, but we believe that this approach can be generally extrapolated to anything pertaining to plant genetic resources. And with this, I thank you very much for your attention.
Okay. So thank you, Ivan. I personally found very interesting this presentation for its applicability to other in situ conservation efforts.
So thanks a lot for the invitation to this meeting, and congratulations for the successful development of the project and the potential infrastructure that will come out. Okay. So I'm going to present the INCREASE. INCREASE is a project, as others presented here, of Age 2020 call about biodiversity, and it's particularly in the same sub-call of the one of Agent and FORGENIUS. We will have an extension of 1 year.
Sorry, you have 9 minutes.
Yeah, I try to go. We will have an extension of 1 year because during the pandemic we have a problem with field trials. And we have 22 partners, most in Europe, and also international partners in Canada and US. And in Europe, we have, in addition to Middle East, like ICARDA, then we have several institutions of GenBanks, IPK, CNRS, in Ra, various gene banks. And also we have ICRIS that is also is international partners. We are focusing on food legumes. And I mean, I think that the first point that we have to start with always is the fact that we have in a totally biodiversity crisis that is also present under the climate change condition that make the things very complex, where in this case, the genetic resources have very important role for conservation of agrobiodiversity, but also for developing new solutions that could be sustainable and give us the chance to cope with these two very big problems. One issue that is the common of these projects that are working on genetic resources, the possibility to enhance the utilization of genetic resources by optimizing the data organization, but also the production of data that can help to explore the phenotypic value and the potential value of genetic resources. Clearly, only phenotyping in these conditions, as this as example, that is also the situation of most of the gene banks, cannot be just sustainable for implement the utilization. So the problem here is of those projects is not only to develop new tools for gene banks, but is also to identify new models of management of genetic resources that will improve the utilization and the innovation pipeline related to the use of genetic resources. We focus on food legumes because food legumes are crucial for all the challenges that we are facing now as a society, and is a crucial aspect to the possibility to move to a new diet that is based mostly on plant-based protein and food. The different crops are the traditional crop, food legumes in the agriculture and cooking tradition of Europe and the Mediterranean, which are chickpea, common bean, lentil, and lupins, with two lupins. One is the Lupinus albus, that is a European crop, but also we include the Lupinus mutabilis, that is from Indian origin. You can find all the information about the project also in a paper that was published in Plant Journal, and you can find this in the site of Plant Journal, but also in the collection of publication that we have in our website, that is pulsesincrease.eu, and we have at the moment 15 papers already published. So this is the concept of the project. The concept of the project is based on nested core collections, based on single seed descent material, somehow similar to what is the precision collection that was previously described by Agent. We have an age core collection that is based on dozen of accession where we have high quality genomes and an extreme level of, let's say, a massive phenotypic analysis at multiple levels with using many tools from molecular phenotyping for, let's say, massive phenotyping in an automatic situation and things like that. So the idea is to have this age core that is very diversified collection of materials, the massive analysis of phenotyping and also of genotyping, including pangenome development. These H-core include also wild materials and unadapted materials, and for this reason, is not overlapping fully with the T-core, which is the training core collection. This base of hundreds, more or less, in general is 400 per species. Accession, always single seed descent, where we have 3 field trials in Europe for 2 years. And so in this case, we have only material that are adapted to the European and Mediterranean environment, so they can grow something somehow in the field. While here we have also exotic material. Here we also employ metabolomics, transcriptomic, and classic phenotyping in the field to characterize these genetic resources for 2 years into location, as I said, and to develop, let's say, genome prediction, and G was the first, but genome prediction and the possibility to find a solution to identify subsect of cell accession that are highly valuable from genetic resources and easily identifiable from the users. A concept of the single seed descent collection, the intelligent collection, I'm describing these 4 papers in current protocols that for each species characterize the materials and tell us what is inside our collections. is the single seed development of increase because it's centralized and is done under insect-free condition. And we call it primary seed increase, while the secondary seed increase can be done in the field only for 1 year, and then is feeding the field trials in all the different locations in Europe. Usually, so basically the project has two pillars. One is the pillar related to the genomic characterization and, let's say, the optimization of the database and practice in the genetic resources context. And the other one is the participatory science and citizen science approach, because our idea is the fact that we need to somehow include innovative way to develop the innovation pipeline in agricultural systems and food system of these genetic resources. So we think that we cannot think that the innovation pipeline is based only on breeders that use genetic resources for develop varieties, but we have to involve the whole society, including the user and the people that transform this the agricultural products in food. So for this reason, we developed this system, and also to let's say, enhance the activity of research in this crop that were abandoned by research for dozens of years, let's say after the Second World War. And so in this way, we want also to develop stakeholders, which means any scientist, any industry, any farmers, any seed company, whoever want to give us a help and then want to be part of the project. They can be part of the project, becoming part of the stakeholder consortium. Now we have more than 150 stakeholders that are different type of different places. Along with, we share the materials that are available and also all the information that we are producing the project while the project is running. This is an example of the interaction with the University of Bonn, that is a stakeholder of us for developing phenotyping approach using drones of the different growth habits of common bean. And these are the collection we have provided. And we, let's say, work closely together for developing this project. The same is happened in chickpea for seed company in Belgium that will develop variety in chickpea or in Belgium and things like that. I'm not going to go, it will be too long. So I want to go here because here is, I think, is one of the aspects that I think is more innovative of our project is the development of the citizen science experiment with the declared objective to develop a decentralized conservation approach to be able to conserve genetic resources in a decentralized and distributed way, in a way that innovation will be easily developing as the material, the genetic resources will be already spread in the different area of production and consumption. So this, I think, is a very important concept, and we really made this project to develop all the tools to make it and to develop exactly a conservation in a, let's say, in a parallel way to conserve genetic resources in based on the participation of citizens. This is the experiment. The experiment is based on an app that we have developed that can be downloaded. You can, let's say, obtain more than, I would say, more than 1,000 varieties, single seed descent lines as usual, and also genotype material from our project. If you request that, you get 5 bags of seeds with different accessions, plus 1 bag of seed that is based on a common variety that will be used as a control. And then you can grow it in your garden, in your field, in your terrace, wherever you want. And this is also, you can find information here in the books, increase.eu dash experiment. And then you can also phenotype it using always the app, and also give us information about the possible use in cooking time, cooking condition, and things like that. We have also a Facebook group of the citizens that participate in this experiment in different languages, because when you do citizen science, you have to deal with the fact that you are going to speak to everybody in their own language. You cannot use only English. This is the participation overall Europe that was, we think, particularly significant because it's more the 16,000 citizens overall between 2021 2023. We have run 3 rounds of citizen science experiment, and in the last round we have about not more than 9,000 citizens registered, and this is the distribution over Europe. Also, this participation is also helped by the stakeholders like the Nordic Gene Bank or the Botanic Garden Maze from Brussels. Another aspect very important is the fact that this system is based on a full traceability of the germplasm, because every movement of seeds within increase, but also within the citizen science experiment, is based on the exchange of standard material transfer agreement. And this is done with the help of the treaty and FAO that are part of the project, so they are member of the consortium. And then this is based on the use of DOI, and also this system is, and that is also coordinated with GLIS, guarantee the full traceability of this exchange of material, because we send the seed to the citizen the seeds, but since last year we also allow citizens to exchange seeds between them, and also in this case, the citizens do that through the app, exchanging easy SMTA and DOI. In this way, all the system of exchange and distribution is able to say what we are conserving, where, and who is going to give something to someone else. In the idea that it's also possible, based on this full traceability, to track any problem that may occur during the conservation and multiplication process. This is the app where we use both, let's say, the possibility to input data for each let's say, traits that can be chosen by the user choosing the skill level, but also we are training the possibility of developing image analysis based on AI, and this is, for example, the example of the picture that the citizen give us. We send to them a color checker and size checker, and they measure the pot not only by telling us the length, but also to making this picture. And this picture, combined to the observation to the citizen and others, we are used to train algorithm to develop image analysis that could be automated. Finally, just to see, are these data somewhat useful or not? Here we see that we can see a very nice distribution of flowering time among the citizens in the first 2 years trials. And we see also that the only one that departs is a material that has been found by genotypic analysis associated to a material that is somehow highly sensitive to photoperiod. Moreover, we tried to do GWAS, and then we were able to perform very well the GWAS, and in a way we obtained high level of heritability, that is more than 60%, and also identify well-known QTL for flowering time in chromosome 1, and this tell us that somehow our analysis is going fine, but also that the citizen give us very good and high quality data as far as we treat it properly. Now we are going to have for the round 3 the data from the satellite data that can tell us on the kilometer square resolution the temperature and the rainfall hourly based, and this will help us also to manage the data set, because each citizen will be let's say located in a precise position, and so we will able to associate to the data of the citizen all the environmental data. This we think that will be very important. This is the core team, where the core is IPK, our group in Ancona, URIS, but also our engineer people that do on the informatic part. Marco Sopperman from IPK, also working on database. Marco Mazzella from FAO. And this is also the work that is a lot of preparing the seeds and send it to everybody overall. So we think that is a picture of the citizen of a school from Portugal that won the video contest that we do every year. and also that we send to them the T-shirt of increase. So we think that our data show clearly the possibility to develop a very high quality decentralized conservation system that can guarantee the full traceability of the seeds and also improve conservation, but more important, can improve, highly improve the utilization of genetic resources worldwide. I go, if I have some more time. Do I have some more time? These are the multi-location field trials. How many? One?
One, yes.
One. Multi-location field trial, some feature that we have produced during the various places in Europe. I'm going back to the first pillar. This also, we do an additional field trial for common bean and maize in intercropping that is coordinated by Maud Tenaillon from CNRS in Paris. And the idea is to identify the basis of the interaction, the genetic basis of the interaction between the two crops to identify the possibility to develop new breeding for intercropping. This is the testing drought condition in IPK using automated system Lemnatec and also in Max Planck Institute in Golm that is doing also the metabolomic data for most of the material. We also work on the nitrogen fixation by, let's say, sampling based on annotation, the genetic, the genes that are important for this interaction and try to identify subset of genotypes that, let's say, maximize diversity for these genes. These are a couple of papers that have been recently published. This is the metabolomic atlas for all the species that we have done as a preliminary step to do all the metabolomic on the field trial. We have developed a new system for genotyping with the work of Marcello Stato, also the group of University of Verona, and in collaboration with Kerstin Beck, that is also part as an international partner of the project. We have developed a new method of depletion for repeated sequence that we use and successfully using for the sequencing of lentil that has a very large genome, and is published in Genome Research. We have developed a common bean pangenome that will be, I hope, submitted very soon for publication. We study the genetic diversity and also the landscape genomics. And then I think that I'm on time. Thank you for listening to me, and if there is any question, I'm happy to answer.
Thank you, Roberto.
Okay, thank you, everybody, and thanks for inviting us here. It's an honor for us to present the project in this public. So I will try to be as brief as possible because of the time. My name is Hristofor from Córdoba University, Spain, and I'm going to present the Gene4Olive project, which is a project about the olive crop. And in a brief, as I say, this project is coordinated by Córdoba University. We are 16 partners from 7 countries. It started in 2020 and it will finish next year in 2024. There is a budget of about 7.5 million euros. And the main objective and topic of this project is pre-breeding activity in olives, mainly phenotyping and other activities that we will see inside of the project. Here participate 5 main germplasm banks of the olives, including Crete, Greece, Morocco, Spain, Turkey, and Italy. So the main objectives of this project are under the same protocols. Firstly, we defined the protocols to work under the same methodologies. We are characterizing and evaluating phenotyping about 600 olive cultivars across the Mediterranean basin in different topics. In the main ones like biotic stresses or climate change, adaptation to mechanization, production, and quality. Then our target is to have a good coordination in preserving these genetic resources and make all this information accessible to the end users and pave the way for the breeding in the next years. So inside of the project, we have had to call for the companies, for the SMEs, and there are about 25 projects founded that has started some breeding lines in olive crop. Okay, this is more or less the scheme of the project. We are working, as I said, firstly, we finished it with the protocols for evaluation. We are working in wild and domesticated olives, cultivars, under firstly making the prospection or collection of the wild cultivars, sampling, and then categorization in base of the different characters. On the other hand, we are developing biomarkers to accelerate the breeding process, and also we are studying the environmental impact on these characters. And in the end, we have a dedicated work package to informatics in order to have a solid database for the olive, in order to give all the information as maximum to the public, and then develop 2 apps based in the artificial intelligence and to the image analysis for cultivar identification and pest and disease identification. We will see in next slide. So I wanted to show some key results that we have obtained in these first 3 years. So some of them, okay, biotic, abiotic stress, agronomic, and olive oil quality. Regarding to the olive oil quality, for example, we have analyzed by the moment 600 cultivars, during 2 years, and some preliminary results. Here we can see, for example, the fatty acid composition, that we find a huge variability in the main fatty acid compounds and, okay, families. If we see the oleic acid, which is the most important one in olive oil, we found that the variability in this big set of samples was from 55 to 85 more or less %. And the media or average was about 72 or 73%. So we can see that we have a lot of resources in the terms of oil quality inside of these germplasm banks in the Mediterranean basin. Okay, then we have classified the olives, ranking them, making some groups in base of these qualities, for example. And also we have found that some of cultivars are fallen out of the standards of IOC standards, International Olive Council standards, just because the cultivar doesn't produce, for example, more than 25% of oleic acid, and this cannot be considered as extra virgin or olive oil. So this project also may affect somehow the quality criteria for classification of the olive oil in this case. The same happens in the sense of, for example, phenolic compounds or oil quality. We found cultivars that has a total phenolic composition at about 0, and cultivars that may have more than 1,500 ppm or milligram per kilograms or phenols per kilogram of oil. So these results explain that it's highly important, the phenotyping, and then go to the next steps, that is the genotyping and other steps. No, but we know that the olive can give us much more benefits than what we have seen till the moment. No, by the moment. Also, we are working and analyzing agronomic characters such as olive yield. So we have some reference cultivars like Picual in Spain, and then we are finding that other cultivars may have higher oil content, which is something that can contribute for the future breeding and improving the breeding of the olive crop in the future. The same for the fruit detachment, for example, for mechanization of this crop, and drought tolerance, which is a big problem by the moment because all of us knows the problems of climate changing and precipitation that are every year less and less. So we are founding cultivars that are not used but are in the germplasm banks and may solve in the future some problems related to the drought and climate change. Also, for the olive table, for example, we are measuring the brushing index, and we found cultivars that are highly resistant, for example, to brushing, which is the bottleneck or the main problem for mechanization of the olive table. Then for the phenology, which is also related to climate change, also we found cultivars that flowers much earlier than the other, for example, no? And we are classifying. Sorry for passing very fast with all these results, but if not, I have to take 2 hours. And then pest and disease, again, fruit fly. Yesterday in Crete, I was talking with some farmers, and here, for example, is a big problem as far as I know. So here are cultivars that looks to be highly resistant, no, to the fruit fly pest, for example, no. So all these results, we think that will have a directly contribution to the farmer, not just publications that are important too. In parallel, we are evaluating the 600 cultivars in about Verticillium, Xylella, Coletotrichum, and Spilosea diseases, and we are having results. Regarding to the environment effect, so these are the 5, let's say, germplasm banks, or the 5 fields that participate in the project, and we can see the water stress of this field is very different. So it makes possible to compare the effect of the different climatic conditions to all the characteristics of olives, development and the oil quality. So in base, we measure it, for example, moisture index, which can indicate us the water stress of the olive. And also we collected all the climatic data, and we found significant differences between banks, between regions such as Morocco, Spain, Greece, and Turkey, and Italy, in bud burst and flowering. And as summary, we saw that about 3 degrees differences may delay the flowering or may accelerate the flowering by 30 days. So 10 degrees may affect like 3 degree may affect like 1 month, no? Or 1 degree may affect like 10 days, accelerating or delay. This is preliminary result, but it is clear that by knowing the classification, we can avoid things that happens this year. For example, that in the pollination of the olive, a very hot event or drought conditions may, let's say, kill the flower or may cancel the pollination of the olive. So it's highly important to these results to know the effect of the temperature and also the solution that we may have having these genetic resources. Regarding to the fatty acid, for example, we didn't find different high differences or significant differences between banks. So it is a character that highly depends to the genotype, to the cultivars, the variants of this character. So it is a positive thing, or okay, we know it that it is not affected. But in the case of the phenols, we found that they are highly affected by the environmental conditions. So these results show us how much can be affected the quality of the oil and also the crop by the condition of the environment. Then next, as I said, we are also considering the wild olives, these resources that can have a really high impact in the future in the breeding of the olives. Firstly, we are sampling in almost all the Mediterranean basin these crops, and we are characterizing them mostly in the most important characters such as diseases, drought, and frost tolerance right now. And also we want to have a conservation, to have a collection of these seeds in order to have then the possibility to have direct access to these resources. Then also very important for the olive crop were the development of functional markers or genetic markers that may accelerate the breeding process, the selection of the cultivars. No? So we are working and having results in biomarkers for selecting cultivars that may be resistant to Xylella and Verticillium, may be resistant to cold and drought, and also some markers that we are really advanced on that may predict the juvenile period of the olive, which means that when we have the first seedling, we may analyze the expression of the microRNA and more or less estimate how much time we will have to wait in order to have the first fruits and the first production. So it helps us in selecting the cultivars in the early phase and make shorter the period that we have to wait for the selection of the new cultivars. Then one of the last points and important point is that, as I said, all this information is getting available for all the public, for all the end users. So with Sapienza University from Rome, Italy, we are working intensively in doing a solid database, a kind of web page that the end user can filter everything for every cultivar that is being evaluated and choose the ones that may have more interest for one player or the other, let's say, you know. So a kind of tool to make accessible easy, to make these resources accessible for the public, but also to be easy to understand for the end user, not too much complicated. On the other hand, as I said, we are working in the 2 apps that we have good results for identification of the cultivars based on the, let's say, endocarps of the olives, and also on the symptoms of the olives for detecting the diseases. Regarding for this work, we are publishing a paper in this journal, and we have had very good results. And the last is that, as I said, we founded 25 projects in total in 2 open calls for the companies, for the public, for the private companies. And this was, we think, a really good point to make the connection with the private sector and to involve them in the breeding and pre-breeding process of the olive. So the next step for us will be the genotyping, because first we needed to phenotype the olive. Goes a bit is less, let's say, develop it in this sense. So firstly we decide to do a solid phenotyping, and now the next steps, our objective is to go through the genotyping. And this is all. Here is the